This is the Senior Design 1 poster for the temperature-controlled whole organ perfusion system project with faculty mentor Dr. Charles Lee. We were allocated a budget of $1,000 in order to tackle the objective and submit the deliverables as shown here. In order to complete this, we took on a systems engineering philosophy of design, in which we started with conceptualization, where we broke down the project into 3 subsystems. Here, the project poster outlines the design phase, and we will move into prototype testing and finalization. Our 3 subsystems are the heat exchanger, the mixing methodology, and the spheroid holding apparatus, connected by the items seen in the Venn diagram.
For the project specifications, the heat exchanger is required to heat the chiller solution from a range of -30 to 0 degrees Celsius using a simplistic PID. The cryoprotectant loading and mixing system is required for the cryoprotectant and buffer solutions to be mixed before entering the sample in one homogeneous solution where it will be loaded in an automated ramped manner. The material in contact with the cryoprotectant solution must not contaminate the sample. The holding apparatus systems must be able to cryopreserve 8 spheroid conical test tubes simultaneously. An image of the system design is shown on the right, demonstrating the chiller fluid entering the heat exchanger, then going through the passive heat exchanger and entering the spheroid holding apparatus. The CPA and buffer pumps solution entered the mixing mechanism and through the passive heat exchanger, then into the liver.
Here are the design details for each subsystem. For the heat exchanger, the chiller fluid is heated by Peltier devices, which control the output fluid temperature. A MOSFET power circuit in parallel is used to control these devices. Multi-channel water blocks create the required extended tube length in a small design package. The mixing device creates centripetal motion to induce mixing of CPA and buffer solutions. This design also maintains incompressible flow. The material consists of temperature, chemical, and wear-resistant 3D printed plastic. The holding apparatus stores up to 8 test tubes. The chiller solution flows through the apparatus to cool these tubes. Images of each component of the system design can be seen below, with notable features highlighted, such as the Peltier devices and water block for the heat exchanger, the removable spiral insert and female lure fittings for the mixing device, and the drop-in test tube holder with accompanying lures.
Here's the overview for our control system for the entire process. As you can see, it needs to be an automated lab view system, and the main feedback points are flow rate, temperature, and concentration. It needs to be a linear and ramped loading and unloading of the cryoprotectant. And as you can see from the block diagram at the bottom, the Fluke VISA thermocouple readings are what drives the other calculations for the rest of the parameters. This slide contains fall 2021 implementation plan. August consists of procuring parts, September is prototype assembly and testing, October prototype adjustment, November finalizing prototype and testing, and December is the expo.